In this video we will discuss about the multiplexed RIBOmap experiment in the HELA cells, and they compared this multiplex experiment with the competitor, that is StarMap. So this is the overall view of how they did their RIBOmap experiment. So what they did is they used 981 gene that are translated in situ within the HELA cells, and they map those 981 genes into various ontological analysis, and same they did with the star map. And how many cells they are able to identify uh, in the RIBOmap is the 1813 cells, and in the HELA the star map uh, technology they are able to identify around 1757 cells. And then they did two kind of things. First one is the co-regulation translation, co-regulated translated proteins that are here, and co-localized translated proteins that are identified here, genes we can say that. So along with that, what they did is they did the few key imaging of the cells in order to identify the cells based on their cell cycle functioning, or at what stage of their cell cycle the particular cell is on. And then they compared it with their analysis results that they get from star map and RIBOmap. This one is the cell cycle assay. From top row is from the we can say RIBOmap, and bottom row is from the star map. Here they identify the cell cycle 1, the maximum signal of the cycle 1, that is shown by RIBOmap here in the top, shown by star map in the bottom. And these three are for the single cell cycle images, and with the zoom in image, as we can see here. And as we go more close, we can see the cells that are labeled with different kind of fluorescence based on their cell cycle stage. And here they have stained the different organelles of the cells using different dyes. That is, DAPI is for the nucleus, flamingo and Corne is for the periphery of the nucleus, or we can say endoplasmic reticulum. So panel C is described the different stages of the cells. The top left described is from the RIBOmap, and this one is described is from the result that you get from few key imaging. And same thing they have done with the star map. This top right is the result they get from star map, and bottom right they got the result from few key imaging. As we can see that both the images are comparable. So in this case, both the RIBOmap and the star map are able to distinguish different stages of the cell cycle. And they did the analysis, single cell translatom analysis, to identify the co-regulated genes. And these genes that are close to the slope are considered as more closely regulated. And from this they have identified 5 kind of gene ontology terms, five gene ontologies that are related to the cell cycle. One that is mentioned on the slope: 1, 2, 3, 4, 5. And among these 5, 2 of them are shown here, that is RTM3 and RTM5 are shown here. And in this case, the genes that are related to cell cycles are highlighted in this heat map. As we can see, are highlighted here, and also their expression labels are highlighted within the heat map. And same thing we can see for RT-web. The genes that are related to G1 and S cycle are highlighted with the pink color, and same has been highlighted within the and same their expression levels are highlighted within the heat map. So this indicates that and these results are shown here for the RIBOmap. This indicates that RIBOmap will be able to distinguish the genes, also distinguish the genes that are involved in the different kind of cell cycles. And from this we can make the prediction or estimation that one of the estimation that is made from this figure uh, is that majority of the translation of the genes are occurred within the uh, G1 and S phase, and it is logically also correct, as in the G1 and S phase the produce uh, cells are trying to grow itself and produce more uh, proteins so that they can prepare themselves for cell division stage. So this conclusion can be interpreted from the results that we get from single cell transcriptome. Later they did the subcellular localization uh, of the cells to identify uh, whether the genes are expressed in a particular organelles or not, and from this they also made the 5 LTMs, which is they indicate the co-localization co-localization uh, proteins. I am not able to recall the exact full form, and they are able to here the results are shown both for the star map and also for the RIBOmap. This one is for the star map left side, and right side is for the RIBOmap. And in this panel, what they did is they are able to identify 5 kind of co-localization gene ontology uh, group, and that the description or bar plot of those groups are shown here. In that case, the LTM1 is for the cell division that they have able to identify. These are the proteins that are involved in cell division or lies or translated in a specific region of the cells, and these kind of things, and the further explanations are also represented here. Later on they shows out of these 5 LTM they shows 2 LTM here, and in this case the 2 LTMs that are shown here are more or less related to the endoplasmic reticulum, that can be further related to the endoplasmic reticulum, or we can say in the periphery of the nucleus. That is further modified, that is further substantiate by the RIBOmap imaging. In this case we can see that the LTM3 genes and LTM4 genes are in the periphery of the nucleus or in the endoplasmic reticulum. And later on they did the quantification of this image. In this we can see that most of the in which we can see that the percentage of the ER localized proteins are around 30% for the LTM3 and around 50% for the LTM4, and for compared to the all genes that they have analyzed, around 35% of them are translated in the endoplasmic reticulum. So ER could be the main machinery or important machinery for the translation of the proteins, as we know that most of the post-translated proteins uh, are either co-translated in the endoplasmic reticulum and then go for protein foldings through the chaperones or with the help of chaperones, and also the initiation of the aniline glycosylation occurs in the endoplasmic reticulum. Then finally what they did is they checked the overlap of these genes within the cells, and or overlap of these genes, uh, and in this overlapping they are able to see that the overlapping of this gene through this data set that are available in the apex. In this they can identify that the all genes are overlapping to around 20%, whereas LTM4 is overlapping to a very great extent, around 90%, whereas LTM3 are overlapping around very less, around 5%, 3 to 4%. So this data set also able to reveal some new finding, as very few LTM3 genes are already known, and very few of the all genes that are present in the endoplasmic reticulum are known to be translated in the endoplasmic reticulum. So from this figure, they are able to made a point that this RIBOmap technology can be used to identify a more stringent mRNA translation location within the cells that have previously not been known. If you need to understand this thing in details, as I'm not able to describe this whole image as clearly as it has been written in the paper. So if you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.